Chemotropes is they use light as an energy source. So those organisms convert those light energy into a chemical energy, which is what we call the photosynthesis. Chemotropes use either anorganic chemicals or either inorganic chemicals for their energy source. For example is the chemolithotropes. So chemolithotropes uses inorganic chemicals as their energy source, while the chemoorganotropes they use organic chemicals for their energy source. Autotrophs use carbon dioxide as their source of carbon. For example is the plants, the algae, and the cyanobacteria. They use carbon dioxide for their source of carbon. Heterotrophs, those organisms that use organic compounds other than carbon dioxide as their carbon source. For example is the human. So they use organic compound and the carbon dioxide for their carbon source. Photoautotrophs is they use light as their energy source and carbon dioxide as their carbon source. So it is the combination of the phototrophs and the autotrophs. Photoheterotrophs they use light as an energy source and organic compounds other than carbon dioxide as the carbon source. So it is the combination of the phototrophs and the heterotrophs. Chemoautotrophs is they use chemicals as an energy source and the carbon dioxide as for their carbon source. Chemoheterotrophs they use chemicals as an energy source and organic compounds other than carbon dioxide as the carbon source. Metabolism is the chemical reactions that occur within any cell. So metabolic reactions is enhanced and regulated by the enzymes, which is the metabolic enzymes. So a certain metabolic reaction can only be performed if it possesses the appropriate metabolic enzymes. A metabolic enzymes can only possess an enzyme if the genome of the cell contains the gene that codes for the production of that enzyme. Biologic catalyst. So these are the enzymes. So the enzyme is an organic compound that acts as a catalyst for biochemical reaction. So catalyst is the substance that decreases the activation of energy for the chemical reaction. So a particular enzyme can only catalyze one particular chemical reaction. Endoenzymes are those that catalyze reaction which remains in the cell. So it happens only in the cells. While the exoenzymes, it catalyzes in the extracellular reactions. Apoenzymes, those that cannot catalyze a chemical reaction on their own. That is why it must link with a cofactor to be able to catalyze. Catabolism is the catabolic reactions which occur in a cell. So it is the breaking down of a large molecules into smaller molecules. It requires the breaking of bonds. So when chemical bonds are broken, the energy is released. So catabolism is the major source of energy. Anabolism is the breaking down of smaller molecules into large molecules. So it requires the formation of bonds. In anabolism, energy is required para ma-form and bonds. So, an energy nga nare-release ang catabolism is ginagamit ang anabolism para makapag-form hin bonds. Biochemical pathways, it is a series of linked biochemical reactions that occur in a stepwise manner. So, biochemical pathways, may daya steps na kailangan sundon from an starting material then to the end product. So, there are two common processes in the biochemical pathways, which are known as the aerobic respiration and the fermentation reactions. So, in aerobic respiration or cellular respiration, there are three phases that occurs. First is the glycolysis. So, glycolysis is a 9-step biochemical pathway that involves the 9 separate biochemical reactions. So, each of which reaction requires a specific enzyme. So, glycolysis can take place in either the presence or absence of oxygen. Kaya ito oxygen, diri na participate in the phase of aerobic respiration. So, glycolysis na produce yan very little energy. Then, glycolysis take place in the cytoplasm of both prokaryotic and eukaryotic cells. Next is the Krebs cycle. Krebs cycle is a biochemical pathway which consists of 8 separate reactions, so each of which is controlled by a different enzyme. So at the end of the 8 reaction, the biochemical pathway ends up back to its starting point. So meaning, after mag-end ang 8 reactions, ang biochemical pathway is magtatapos lagi hapiya as a starting point. That is why it is called as cycle. While the electron transport chain na consist iyahin series of oxidation reduction reactions. So, din yung electron transport chain, din na-produce and more energy. Then, the energy is released as electrons are transferred from one compound to another. Next process is the fermentation reaction. So in this process, they do not involve oxygen. Therefore, this fermentation takes place in anaerobic environments. So on first step, fermentation reaction is glycolysis. So we all know that glycolysis do not involve oxygen and they produce very little energy. The next step is the fermentation reaction is the conversion of pyruvic acid into an end product. So, on particular end product na produce is na depende on specific organism that is involved. So, those various end products of fermentations have many industrial applications. For example, a certain yeast and bacteria is converted into pyruvic acid, into ethyl alcohol, and carbon dioxide. So those teas are used to make wines, beers, and other alcoholic beverages and bread. Then the oxidation reduction reactions. So this oxidation reduction or redox are paired reactions in which the electrons are transferred from one compound to another. So, when an electron or an atom or ion molecule loses one or more electrons, it is called oxidation. Then resulting gain of one or more electrons by a molecule is reduction. But reducing agent is the electron donor, while an oxidizing agent is electron acceptor. Mutation is the change in the characteristics of a cell which is caused by a change in the DNA molecule. So, ibig sabihin, if magkamay the change sa DNA molecule, ang characteristic ng cell is magkakamay the gap changes. So it is transmissible to the offspring. There are categories of a mutation. First is the beneficial mutation. It benefits the organism. It enables the organisms to survive in an environment, whereas those organisms without mutation, they would die. And it also enables the organisms to be resistant to a particular antibiotic. While the harmful mutation, it leads to the production of a non-functional enzyme. So those non-functional enzyme enables to catalyze the chemical reaction. And lastly is the silent mutation. These mutations have no effect on the cell, so they cause no change in the function. Mutant are those organisms that contains the mutation, while mutagens are those agents such as the physical or the chemical agents which affects the chromosomes, and it causes the mutation. Here are some following ways in which bacteria can acquire new genetic information, such as the lysogenic conversion, transduction, transformation, and the conjugation. Lysogenic conversion. So we have two categories of the bacteriophages. By the way, bacteriophages are those virus that infects the bacteria. The two categories of a bacteriophages are the virulent bacteriophages and the temperate bacteriophages. Virulent bacteriophages are those bacteriophages that always cause the lytic cycle to occur, while the temperate bacteriophages are those bacteriophages whose DNA integrates into the bacterial chromosomes, but it does not cause the lytic cycle to occur. And for your information, lytic cycle is the sequence of events in the multiplication of a virulent bacteriophages, which ends with the lysis of the bacterial cell. Then let's proceed to the lysogeny. So lysogeny is when the bacteriophage's DNA is integrated into the bacterial chromosome. So an bacteriophage's DNA replicates along with the chromosomes. And the prophage, it is the name given to the bacteriophage when all that remains of it is its DNA. It is integrated into bacterial chromosome. So, the lysogenic conversion happens if the prophage is integrated into the bacterial chromosomes. The bacterial cell can produce gene products that are coded for by the prophagenes. So, it does not usually cause the lytic cycle to occur, but there are certain events that can trigger to do so. So, an lysogenic conversion, in other words, an bacterial cell has been converted as a result of the lysogeny, and it is now able to produce one or more gene products. Next is the transduction. It involves bacteriophages. It means to carry across. So, some bacterial genetic material may be carried across from one bacteria cell to another by a bacterial virus. So, this occurs after the infection of a bacterial cell by the temperate bacteriophages. Another one is the transformation. The bacterial cell becomes genetically transformed after the uptake of the DNA fragments, or also known as the naked DNA, from the environment. And lastly is the conjugation. So it involves a specialized type of pillows which is called the sex pillows. So an donor cell and the recipient cell of the bacterial cell is attached by the means of the sex pillows. Some genetic material are usually in the form of a plasmid. Conjugation involves with the process of a bacterial mating, and it uses the terms male and female cells as a reference for the donor and the recipient cells. Plasmid is an extra chromosomal DNA. So if a plasmid na contain multiple genes for the antibiotic resistance, the plasmid is considered as the resistance factor or the R factor. And then if the recipient cell who receives an R factor becomes a multiply drug resistant organism, it is referred to as a superbug. Genetic engineering involves the introduction of new genes into a cell. When a cell receives a new gene, it can produce the gene product that is coded for that gene. While gene therapy involves the use of viruses and plasmids to introduce normal genes into cells that contains abnormal genes.